My name is Laurent Francioli. I am a postdoc in Daniel MacArthur Lab at Massachusetts General Hospital and at the Broad Institute. My main work has been on the Genome Aggregation Database, or GNOMAD, which is the largest public database of exomes and genomes. It contains 123,000 exomes and 15,000 genomes. And right now, this is accessible for the public through a web browser. But for internal use, we want to be able to query these data in a flexible and scalable way. To do this, we are using several different technologies depending on the type of query that we need to run and depending on the people who are running these queries. And one of these technologies is BigQuery. And I will here present what BigQuery is and how and why we're using it in the context of the GNOMAD project. All right. So BigQuery is a service provided by Google, and it is a fully managed enterprise data warehouse. What does this mean? It means that you can store all of your data in table formats. It handles incredibly large amounts of data. In our case, we have about 1 terabyte of data that is stored using this service. But there are cases where hundreds of terabytes or petabytes of data are being stored and queried using this service. It is fully managed, which means that you don't have to worry at all about what is going on behind the scenes and how the magic happens that you can query such a large amount of data. You do not have to worry about authentication. This is taken care of via the Google authentication. And you never have to worry about updates to the product or anything. Everything is managed for you. So to dig a little bit more into what the product is, it is a distributed column store. This means that once you upload your data, and you can, there are multiple ways of uploading the data, but essentially everything is represented as tables, and so typically you will upload your data as tabular data. Your data is then distributed or sharded into multiple small pieces that will be put in different Google data warehouses where they have compute power that you can use. It is a column store, which means that the columns of your data are stored. The values of each column is stored together, unlike your typical database where each row is stored together. This has implications that I'll discuss a little bit more later. And it allows fast queries on these data using the SQL, the SQL language. And so here I've put fast in quotes because it really depends on how much data you have in some sense. And it will be very fast for a very large amount of data relatively, but it might be a little slow for a small amount of data. There is a lot of overhead in how BigQuery works. Essentially, when you start a SQL query with BigQuery, it has to find a number of computers that are close to your data, start a BigQuery process on them, and then go through your data. And BigQuery doesn't have any index on the data, so it will always go through the entire data sets in order to apply the filters or aggregations that you want to do on your data. So the type of times that the type of time that it takes to query the data is typically seconds to minutes. So in the case of a petabyte of data, this might be considered very fast. In case you have only, say, 1 meg, this is going to be considered very slow. So it all depends on the amount of data you have and the type of response you need to have. It can be accessed through a web UI, and I'll show this. There's a REST API. There are several client libraries in Python, in Java, in R. I'll very briefly flick through what the same query looks in R, just to have an idea. And it's integrated within the Google Cloud environment, so it's fully integrated with the authentication. It has bridges with Google Drive, Google Data Studio, and so on. Let's look at our case scenario, which is querying Nomad with BigQuery. Here, our use case is that as part of the internal version of Nomad, we have the genotypes of all of the samples that are in Nomad. Now, we cannot release these genotypes publicly, but internally, we can query them in certain cases for certain studies. Now, some of the analysts that are working, for example, on rare disease patients, find a variant that is also in Nomad, and they want to know in which samples they are in Nomad, and which are the genotypes of these samples, whether they're in cohorts that they could recontact to eventually maybe do further phenotyping, and so on. And so these people are analysts that are not bioinformaticians, and so they needed a simple interface to query these data. Because these data are very large, it's impractical to put them in a regular database, and so you have to go through a distributed system. So here you have multiple choices. You could have a system that you manage yourself, where you can tune the performance, have indices, so you could do use something like Elasticsearch. But this means that you have essentially, if you want this service to be available 24/7 to analysts without them doing anything, it means you have to manage it for them, and you have to have a number of machines and hard drives that are working for you 24/7. In the case of BigQuery, you can essentially just put your data in there, and they have a ready-to-use web UI, and if needed, bridges with other programming languages. So the data needed to go into this database, we have essentially the data that is in the site's VCF. So this is information about variants. So you have chromosome position, reference allele, alternate allele, the allele frequency, the allele count, and so on. All of the QC metrics as well. So this is a few hundred fields. Then there are the genotypes. So again, chromosome position, reference, non-reference allele, and then the sample name, the genotype, and also the genotype quality, the depth for each allele, and so on. So some QC metrics. Then we have a table of sample metadata where we have, again, the sample name, the population, whether the sample would be released as part of the public, whether the sample can be recontacted, and so on. And then we finally have a table with transcripts that were annotated through the variant effect predictor program, where you have, again, chromosome position, ref, and alt, and then the transcript, the consequence, and then all of the web annotations. And so the way BigQuery works is basically you upload these tables independently, and unlike traditional databases, you do not have to specify any keys or any specific relationship between these tables, because the tables are not indexed, and so the joins and other filterings can be applied to any two tables. You just have to specify which fields you want to join on or you want to filter on. Now, of course, these are four different tables, and the analysts that are going to do this aren't SQL experts. So in order to give an easier review of these data, we also created a view on the data that is essentially performing for you the join on the variants, the genotypes, and the samples metadata. And we also have one that has the transcript. This allows an analyst to view all of these data as a single flat table, but at the same time to store it in an efficient way, because you're not repeating for each genotype at each variant all of the variant information and all of the sample information. So this is a way where we can both have a very easy way to query the data and an efficient way of storing it. So I'll quickly go through the pricing to just give you an idea of how much this costs, and then I will show you the web UI and how it looks. So in terms of pricing, storage is 2 cents a month per month, and then after 90 days, if you haven't touched the data, so if as in you haven't updated the data, you haven't inserted any more data, the price halves. And then the other part that is important is the queries. They cost $5 per terabyte queries, and the first terabyte per month is free. And so I'll go a little bit more into how this is computed when you're actually doing queries. So let's look at our data that we're storing in there. We have here, in terms of variant information, for the genomes, the coding part is 20 gigs, the non-coding part is 100 gigs, and then for the exomes, the coding part is about 10 gigs. Well, I mean, I guess there's only coding. So it costs us about $1.30 a month. And then for the genotype, so there's here one entry per variant per non-reference genotype. Here we took the part that we only wanted the non-reference genotypes, simply because the use case we have with the analysts we have aren't interested in the samples that are reference, that have reference genotypes. Of course, this allows us to compress the data massively because most of the sample genotypes are reference genotypes. And so this table is only 50 gigs for genomes coding, 250 for the non-coding part, and then 40 gigs for the exomes. And so that sums up to about $3.40 a month. So in total, we were paying about $5 a month in terms of storage. Which for this amount of data is something I think fairly reasonable. Now, how about queries? So the worst possible query, there are 2 extra tables that I didn't mention because they are so small that they're essentially free. The query cost now. So the worst possible query you can do is if you query all of the columns of all of the tables, and then that would be a query that would cost you about $2.40. That said, when you're querying the data, it only looks at the data in the columns that you're actually interested in. So if you don't select all of the data, you don't pay for querying all of the data. And of course, it's pretty hard to know, a priori before you're going to run a query, how much exactly of the data it is going to go through. But to give you a realistic example, if you count the number of variants by population, by data set, so exomes and genomes, and by web consequence encoding regions, this costs about 15 cents. And so this would go through columns, this would go through the chromosome position, ref, and alt columns in all of the tables in terms of doing the joins. And then you will need the sample here to do the join on the sample metadata. And you will read essentially, the consequence from the variants, and then the population from the other table. And so if you are writing this query, across the three tables, this would cost about 15 cents. The first $5 of query per month are free. So it also depends, the pricing and the use case for BigQuery also depends how often you're querying your data. If it's something that you need, that you don't query very often, but that you need to be, but that you don't want any overhead whenever you want to query it, this can be a very cost-effective system. All right. So now I'm going to take a few minutes to show you exactly how it looks in the browser. So if you're familiar with the Google Cloud Platform, this is a Google Cloud environment. And here, if you look at product and services and scroll down to the big data section, here there is a BigQuery section that will bring you here to this page. there, there are several panels here. Here on the left-hand side, you have your projects, and the data is organized by data sets. So there are essentially two layers of organization here, data sets. So here I have a data set that's Nomad, and then here under the data set, you have all of your tables and views. And so if I look at one of the tables, say for example, exome genotype, I can see here all of the fields that are here, the type of these fields. I can have a preview of the table, so I can have a quick look at what this table looks like, and I can look at details. And here in the details, I can put a little description of the table. In the schema, I can also describe what each and every field is. So if you wanted to make a really nice, very clear table that's self-descriptive, this is something that is supported through this platform. And then the second part is here, those that have the green icon are the views. And so if I look at one view, for example, here, exomes, that essentially is all of the data from the exomes. I can see all of the fields that it contains. So this is a view that essentially takes multiple tables and views them as one single flat table. And then if I want to see exactly what is happening behind the scene, if I click on details, I can see the exact SQL query that is used to produce this view. And so this is essentially just the join on multiple tables that are viewed as a view. So what if you want to do a query? Here, there is a little panel where you can write a query on any of the tables. Here, this is the query that I've described before. I want to point out that the interface for querying does support things like autocomplete. So for example, if I start with an M, I can look at what fields are available. Like this, and it also supports. So here I'm using the table Nomad all coding, so I'm using this table here. It also supports here clicking on this table to add fields. So if you're browsing and you want to select a bunch of columns, you can just go here and click on the columns that you want, or you can use autocomplete. It's a fairly convenient interface. Let's now run the query. So here, when the query is running, you can see here for how long it's been running. Here, on purpose, I'm querying all coding, which is the table that summarizes all of the data for all of the coding regions of Nomad. So this is querying right now. The table in total is I think is about 200 or 300 gigs, and the columns that I've queried amount to here 33 gigs. And here you get your results. It's quite nice. You can save the results to Google Sheets if they aren't too large. You can save them as a table here. If you're using Chrome, you can download them as CSV. I'm using Safari, so this doesn't work here. So it's easy to export your table. You can view your table as a table or as JSON, so you can also export it as JSON if that's a better format for you. It has a pretty good bridge between the different platforms. Another thing I should mention is that you can have tables that are actually linked with the Google Sheets. Essentially, the table is a view on the Google Sheet. And so if you modify fields in the Google Sheets, this will be automatically propagated into your database, which might be helpful to bridge the gap between people who are used to store things in a Sheets format and people who are using databases and querying them. So instead of having to reload different versions of the data, this is dynamically done for you. Very quickly, if I wanted to do this query in R, this is what it would take. So you basically load the BigQuery library. Then you have to set the projects. So here, this is the Google project, the name of the project, and then you simply here put the SQL. So that would be the exact same here. You could just copy here and then paste it there. And don't worry about the standard SQL. This is just BigQuery supports different SQL languages, and so this is just standard SQL language. And you can just run this. So basically, you execute the query, you give the SQL that you want to execute the project, and then this is just which SQL language you want to use. So that would be the way of querying this data via R. There's also a Python interface, a Python API, Java API, and one more thing I wanted to quickly point out is that this is also integrated with other Google products. One of them is Data Studio, that essentially lets you plot, data. It's really meant for creating reports. But so here, just as a toy example, I've plotted the counts of the annotation. So here we have missense variants, regulatory regions, and so on. Split by exomes and genome, and here I've put a little filter on the population, and so this is the edit mode where you can add things, but then you can share this, and you can share this in view mode, and then in view mode, people can use these controls. For example, here, if I wanted to see this only on, say, I don't know, the non-Finnish Europeans, I would click this, and this would refresh. Again, this is querying, in the background, the hundreds of gigs of data, and so all things considered, it's not super fast, but it's not super slow either. So it's going to take a little bit to refresh. All right. This is usually slower. There you go. And so now this is only non-Finnish Europeans, and you can hover over things. You can filter the data. You can put different types of graphs. So this might be useful. I'm not sure. It might be useful for your use case. What we have been using mostly is really here, that interface, and what our analysts wanted to do is essentially just download their table as, download their tables, and then and then identify these people. All right. So this is all I had for today. I hope this was helpful. And oh yeah, I should mention maybe one more thing is that BigQuery is integrated with Google Genomics, and so you can also import your data directly from a VCF into BigQuery, and then do essentially all of your analysis in this framework. What I will point out is that the SQL language is a little limited when it comes to performing all of a typical analysis pipeline, but of course it'll depend on the type of analysis that you'll be running. But you cannot do things like regression, for example, through SQL. So it's likely that it won't be enough for doing all of your for really going from A to Z, but it might be enough to do something like import of your data, do some basic filtering, and then aggregation and export this into R, for example. That might be a use case where really going through the Google Genomics pipeline might be helpful. Okay. All right. So that was it. I hope this was helpful. And thanks for watching.